Thank you. Thank you for this wonderful invitation and introduction. I'm so excited to be with you here today and talk about one of my favorite topics. In my research, my main research focus over the past 6 years has been on Picophytoplankton. And what I will talk to you today is a little bit of background to Picophytoplankton, and then I will also go a little bit more into the research projects that we are doing in investigating different controls on Picophytoplankton and what determines their distribution across the world. I will also talk about how our ocean is changing and what this means for Picophytoplankton in the future ocean. Let's start with a tour into space and looking at our wonderful Earth from space. This is a video that you will see that is from NASA, looking at the chlorophyll content in our ocean. So this is the chlorophyll A concentration. And as you can see on the scale, it goes from the purple bluish color all the way to dark red color. And when we travel here around our fantastic planet Earth, you can see that these colors are changing, and there's different areas of the oceans where production is higher than others. So there's a higher chlorophyll concentration, typically around the coastal areas. You can also see around the equator, there's this light blue line where there's a slightly higher productivity. But one of the things that you also notice is that most of the ocean, the large areas of the ocean, are actually this large, darker blue color. And that means that most of our oceans are actually quite low productivity regions. And if we want to make a connection to habitats on land, this is perhaps the deserts. So the large areas of the oceans are really what corresponding to deserts on land where there's low nutrients. And in these environments, it's very harsh conditions. So the organisms that live there have to be specialized to life in low nutrient environments. And this is something that I want you to keep in mind when I talk about Picophytoplankton that are very small in size. So when we think about phytoplankton, one of the things that researchers have done is to divide plankton into different size classes. And this is in order to get a better understanding of the different roles that different plankton species play in the oceans. So the larger phytoplankton is what we typically think mostly about when we go swimming or snorkeling at a beach on a sunny, wonderful day. So these are the large macroalgae, macroplankton that we see with our eyes. And then there's smaller size classes like mesoplankton and microplankton. And these are size classes that we could typically capture using a plankton net. So on the picture here, there's a picture of a multi-net, which is a phytoplankton net that contains multiple plankton nets that can be closed at different depths to collect phytoplankton from different depths. But what most of us don't think about when we go swimming is that the majority of the phytoplankton are actually very small. So they belong to the microbial fraction. And these are the nanoplankton and the picoplankton, and these are really the smallest phytoplankton. But I will try to convince you today during this lecture that we should care much more about them because since they are so many, they actually make a very, very large difference in marine ecosystems and are really the foundations of life here on Earth. So picophytoplankton, they're very, very small. Just like other size classes of phytoplankton, it's a definition that scientists have constructed. And Picophytoplankton are usually defined as being less than 2 micrometer in diameter. And this group is composed both of Picocyanobacteria, so Prochlorococcus and Synechococcus are the most abundant phototrophic organisms on Earth and very widespread. And the Picophytoplankton group also consists of picoeukaryotes, and these are a diverse group of small eukaryotic organisms that are distributed all over the diversity of the eukaryote tree. Here is a microscope image of Baltic Sea Picophytoplankton. And you can see that there's some different sizes and colors here. And this is an epifluorescence microscopy image, and we have done an overlay with different filters. And these different filters, they can show the different pigments that these Picophytoplankton have. And with the size and the pigments, we can distinguish these different groups of small Picocyanobacteria and picoeukaryotes. When it comes to the distribution patterns, here are a few images showing the distribution of Prochlorococcus, Synechococcus, and picoeukaryotes across the world. And I think I can convince you with these images that these organisms are really of global importance. If we start with Prochlorococcus, Prochlorococcus is the smallest photosynthesizer on Earth and also the most abundant. And it likes it warm. It has a wide geographic distribution, but it really is limited to around 40 degrees north and 40 degrees south. And that is because it has a temperature optimum between 10 and 33 degrees Celsius. So this is really what determines the distribution of Prochlorococcus. But it's really highly abundant, up to 100,000 cells per milliliter. And that's a lot of cells. So even though they're very, very tiny, they can really make a difference and contribute to primary production on a global scale. These groups together contribute to around 19% of primary production on a global scale. If we move on to the slightly bigger and a cousin of Prochlorococcus, so these are quite closely related organisms, but they are genetically different. And Synechococcus is slightly larger than Prochlorococcus. It also has a larger distribution, expanding also into colder and more nutrient-rich environments. We can also find it in estuarine and freshwater environments. But as you see on the color scale, it does not reach as high abundances as Prochlorococcus. So Synechococcus typically we find between 10 and 50,000 cells per milliliter. So slightly less abundant, but it also has a larger distribution. When it then comes to the distribution of picoeukaryotes, picoeukaryotes really have a global distribution, but they are considered most abundant in coastal and upwelling zones. And there's usually a peak in the biomass of picoeukaryotes at the deep chlorophyll max. And what I will come back to later is also that picoeukaryotes have a distribution also in polar areas, where we typically do not find the picocyanos. So the distribution of different groups of phytoplankton, it's largely determined by temperature. And in these graphs, you can see how the niche partitioning between the different groups of Picophytoplankton is distributed with light and temperature. And when it comes to the contribution to the total Picophytoplankton biomass, there's a shift where in colder environments, so less than 5 degrees, there's a complete dominance of picoeukaryotes. When it gets slightly warmer, Synechococcus contribution is increasing. And then after 10 degrees Celsius, and Prochlorococcus are also present. So the contribution to the Picophytoplankton biomass is typically determined by the temperature in the environment. There's also a large diversity within these different groups, especially when it comes to the Picocyanobacteria. One of the things that have been widely studied is the different pigment composition of Synechococcus. So the group Synechococcus has really extensive physiological diversity and a large variation in accessory pigments. So the bottles that you see here on the left are cultures that I keep in my lab. They are isolates from the Baltic Sea. And these are all classified as Synechococcus. And when you look at this image, it's almost like in a candy store, right? There's some dark red, there's some that look almost blue, there's some dark green. It really looks like different types of organisms, but they're very closely genetically related. But because of the composition of different accessory pigments, they have these different colorations. So why could it be important to have different types of colors? What could the advantage be of having different types of colors? Well, one of the reasons that we see is that some of these strains are more adapted to life in turbid environments, where the light conditions are very different compared to the open ocean. And if you have visited different regions of the world, you have perhaps experienced that water can have very many different colors. So in some areas, the water can be really, really dark blue. And out here in the Baltic, next to Kalmar, where I live, the watercolor is typically very, very green, especially in coastal environments. So these different strains are adapted to different light conditions in the water, but they often coexist. So when conditions change in the water, one can have an advantage compared to the other. And one of these accessory pigments that we often talk about when it comes to Synechococcus is phycoerythrin. So phycoerythrin is what give these cultures to the left this red color. And this pigment, it's really good in these very clear blue waters. While on the other hand, phycocyanin, which gives this more greenish, bluish color, it's more common in turbid waters. So there's different environmental niches for these different types of Picocyanobacteria. So Picophytoplankton are very, very tiny. One of the advantages of being very tiny is that you don't need very much energy. You can replicate quite efficiently. And this is one of the reasons why they are so successful in these low nutrient environments. So they have a low surface to volume ratio, which also makes them competitive when it comes to acquisition of nutrients. So compared to larger phytoplankton, they are very efficient and do not need as much energy. And this is one of the reasons why they have then a cosmopolitan distribution, where they are found both in marine, freshwater, estuarine environments. They are very adapted to changing conditions. Picophytoplankton are also very diverse, so they are adapted to life in many different types of environments. And because they are so successful, since they have this successful strategy, even though they are so tiny, they are important for many aquatic ecosystems. But in many areas, they are very understudied. And their small size is one of the reasons for this. So the phytoplankton net that I showed you earlier, the picophytoplankton will pass right through the net. We will not even catch it in that type of net. The microscopy methods that we typically use for phytoplankton identification, these cells are too small to be counted as part of the monitoring programs of phytoplankton. So it was not until techniques such as flow cytometry and molecular techniques that we really started to gain a better understanding of picophytoplankton and how abundant and important they are for marine ecosystems. And over the past 6 years, I have focused my research on picophytoplankton in the Baltic Sea. And here in the Baltic Sea, picophytoplankton have not been included in the regular monitoring programs, except for a few stations in the Bothnian Bay where they have been counted using epifluorescence microscopy. And one of the reasons why I think that picophytoplankton in the Baltic Sea are really, really interesting is that we have a very diverse composition of picophytoplankton here in the Baltic. And one of the reasons for this is that we have many different types of gradients. So in the Baltic Sea, there is a salinity gradient from highly saline water coming in through the Danish Straits to the Bothnian Bay, where there's a very low salinity. There is a difference also in nutrients. There is a high nutrient load from land, from farmland, is an area that is very highly influenced by anthropogenic impacts. While in the summer, out in the Baltic Sea proper, phytoplankton growth are nitrogen limited. So there's a sharp nutrient gradient from the coastal areas to the offshore areas in the Baltic. We are also in a temperate region, which means that we have seasonal shifts. And this also affects the succession and the dynamics of phytoplankton. So with these shifting conditions, we have a highly diverse population of picophytoplankton. And there are also shifts in ecotypes as the environmental conditions are changing over the year. And this is one of the reasons why I think this is a very interesting place for studying picophytoplankton and how they are responding to different environmental conditions. So one of the first things that we set out to do was to go sampling, to look at picophytoplankton abundance, diversity, and also to build a library of diverse isolates of picophytoplankton. And here are some of the different sites where I have been working during the past year. So we have conducted a study in the Gulmar Fjord on the west coast. We have also had a coastal and an offshore sampling station here close to Kalmar with the case station, which, as you see on the picture, is located just across from the Kalmar Castle. If you have been here, it's a bathhouse that is located just here, 100 meters from our university campus. And we can go there, and it's about three 3 meters in depth, and it's in Kalmar Sound. But there's an inflow of water in this area. So there's quite high circulation of water, but it still has a characteristic of this very coastal site with influence from land. Our other sampling site is the Linnaeus Microbial Observatory, which is located around 11 kilometers off the coast of the island of Öland, outside of Kårehamn. And here, the LMO is just really a GPS spot that we go to. But we used to have a buoy. You might see a little yellow spot here on the picture. That marine monitoring buoy that we had a few years ago that would measure salinity and temperature and turbidity in the water. It's not there anymore. It drifted away quite a few too many times with autumn storms, and then our technicians got tired of trying to find it when it had drifted out to sea. This monitoring station we sample every other week, and we have done so since 2011. So this is, I know you have heard about time series this morning. I would not claim that this is our equivalent to Station Aloha, but at least we are dreaming of it. So that is perhaps our vision and inspiration, having a microbial observatory here in the Baltic Sea. So one of the ways we can look into diversity of picocyanobacteria is by molecular techniques. So in this study that we did in the Gullmar Fjord, we amplified using PCR a region of the 16S rRNA gene and looked at what different types of Synechococcus were present in the Gullmar across different seasons. And this is starting in February, going over spring, summer, and then autumn, and again January, March. And what we found in this study was that previously it had been reported that the community shifts of picocyanobacteria were really determined by the salinity gradient. But on this scale in the Gullmar Fjord, we could not find the pattern of salinity. Instead, we saw that it was largely temperature and different conditions and stratification during these different seasons that was causing the community shifts in Synechococcus. And we found abundances of almost 10 to the 5th cells per milliliter. So this was a highly abundant group in the Gullmar Fjord. And what these different colors show you on the graph is how the contribution of different types of Synechococcus was during the different years. So you can see quite clearly that in summer, it was this blue and the red sequences that were completely dominant. But during other times of the year, there were different strains that were present. And if we move then into the Baltic Sea and our case station and the Linnaeus Microbial Observatory, what we have done is to divide Synechococcus into two different functional groups based on the pigment. And here, the red dots are showing the phycoerythrin-rich Synechococcus. So the ones that were pinkish in color or reddish in color that you saw on the picture earlier. And the light blue are the phycocyanin-rich Synechococcus, which were green in color. And we looked then over 3 years what the abundance patterns were like. Here at the coastal station, you can see that the abundance of the red and the green Synechococcus were almost as high a lot of the times of the year. So in the more coastal environment where there's higher turbidity, there was also higher abundance of these green Synechococcus strains. If we compare it to the offshore environment, you can see that these red strains are higher in abundance. So those were more optimized to life in the more offshore environment where there's more clear water conditions. And we found that the abundances of the Synechococcus were highly correlated with temperature and nutrients. So when nitrate was low, Synechococcus abundance was higher. And it's also these abundance peaks. Here in black is the temperature. And you can see that these abundance peaks in the Synechococcus populations, it's very well fits with the temperature. And we were also monitoring the pico-eukaryotes, so the PPE populations. And this has not been done very extensively in the Baltic at all. And what we found that the pico-eukaryotes, they were also quite abundant at 10 to the 4 cells per milliliter. They were also peaking in summer where temperatures were around 20 degrees or above. But what was very interesting was when we were comparing the biomass. So if you remember, the pico-eukaryotes are typically larger than the picocyanobacteria. And when we calculate this into biomass, what you see here on the bottom is the percent contribution to the total phytoplankton community. And here the red is the phycoerythrin-rich, the blue is the phycocyan-rich, and this green color are the pico-eukaryotes. And the purple is the other phytoplankton. And what was really almost surprising to us when we were doing these calculations was that frequently the Picophytoplankton contribution to the total biomass here in the Baltic Sea was more than 50%. So that's a quite substantial contribution to the phytoplankton biomass. And it's not only that I'm particularly interested in the Picophytoplankton, but now I started thinking about all kinds of questions. Well, what does that mean? What happens to these small cells? What are they eaten? Are they dying? Are they recirculated? Can they be exported from the water column, or are they too small? Are they too small to be eaten by larger fish? Are they only grazed by nanoflagellates? All kinds of questions started flourishing from this data, because so if it's 50% of the phytoplankton biomass, then we have to know, where does that end up? Does it move up into the food chain, and or is it recirculated? So with this first monitoring of Picophytoplankton in the Baltic Sea, I then started to explore more about what is it that is controlling these abundances. And when we think about phytoplankton growth, we can think about the availability of light, temperature optimum for growth conditions, the availability of nutrients. But then there's also another thing to think about, and that is how are they dying? Are they being eaten? Is it transported to higher trophic levels, or can it be that they were under high viral pressure, so that a large fraction of them actually die and then get recycled into the microbial loop? So these were new areas that I I started to explore. We're not only looking at how abundant they are and how diverse are they, but where does this biomass end up? So when are they growing and when are they dying? And to do this, we do different types of manipulation experiments. So this can range from smaller bottle incubation experiments that we do in the lab, where we have controlled temperature and light conditions. So here is in our aquatic lab, where we have a running inflow of seawater to keep it at close to in situ temperature. And then we can manipulate cultures or seawater with different nutrients. We also have these in situ incubation arrays where we can perform experiments at sea. And to here you really have the daylight cycle, and you have the actual temperatures. There's also a bit of a turbulence in the water as the currents are moving these bottles. So there's a natural mixing in these conditions. I also, for the Arctic expedition, we built these incubators, which are really sturdy, built for the Arctic environment, where we can have an inflow of seawater to keep it at in situ temperatures. So with these manipulation experiments, it's possible then to in the lab manipulate temperature, light, salinity, to then study rates of primary production, growth rates, or pigment composition. So I'm going to speed up a little just so we get, because I had half an hour, Rachel. Is that?
Oh, you can continue. That's fine.
Okay. So one of the things that we wanted to do was look at these co-occurring Synechococcus to try to identify if there were different ecotypes. So if there were certain strains that were adapted to a specific environment, to a certain niche. We had already seen that the different composition in pigments determine their composition in nature, but we were curious about if there were different optima for different strains. So my former postdoc, Annabella Aguilera, has performed a large amount of experiments with these cultures to look at different salinity conditions, different temperature conditions, and different light conditions to see if there's different optima for these different strains. And what the take-home message in this picture is that not one of these strains look the same when it comes to the curves and looking at the optimal conditions. And with this knowledge, it really makes a lot of sense that there's different strains that can co-occur in the environment, because one of the strains might have an optimum at higher temperatures, while one is in fact largely inhibited by higher temperatures. And salinity conditions, some had very large adaptability to different salinity conditions, but some really had optima at specific salinity conditions. And same when it came to light. For some strains, low light did not inhibit growth at all, while for others, they really required high light conditions in order to grow optimally. During the year, we also explored what nutrients were limiting to Picocyanobacteria. And we found that Picocyanobacteria could be nitrogen limited in the natural environment. And we also found that ammonium was a preferred nitrogen source at higher temperatures. And this was very interesting to me because I have a background in nitrogen fixation research. And this could suggest that there's a tight coupling between the filamentous nitrogen-fixing cyanobacteria and the picocyanobacteria during summer in the Baltic, where nitrogen-fixing filamentous cyanobacteria might provide ammonium to these smaller cyanobacteria in the environment. And this is also then co-occurring with these peaks in picocyanobacterial abundance. So this is something that I want to explore a little bit more. We have also done manipulation experiments to look at the grazing rates of Picophytoplankton. So coming back to how much of its carbon is transported into the food chain. And these manipulation experiments, we have done it with having a grazer-free treatment and then a control with grazers. And when we calculate the carbon flow rate, we see that it was quite similar between the pico-eukaryotes and the Synechococcus, but there was a higher grazing pressure on the pico-eukaryotes. So we think that the pico-eukaryotes could be very important when it comes to transfer of carbon in the food chain in the Baltic Sea. This is a study that is still in progress. And the carbon transfer was quite similar for the two different groups. So we have established that picophytoplankton are important, and we have started to explore different factors that are controlling their abundance in the environment and the carbon flow. But optimally, what we want to be able to do is to make predictions about the future. So what will happen in the future when the oceans are changing? How will food chains be affected? And what will happen to this picophytoplankton fraction? Global warming. Looking at these graphs, which show the dramatic increase of the temperature across the planet, it's quite, I have so mixed feelings about this. As a scientist, I'm quite fascinated about these rapid changes, but it's also a development that we absolutely cannot ignore. And this is one of the reasons that I came into looking at the Arctic and the very rapid changes that are now occurring in the Arctic in this very vulnerable environment. And on the graph to the right, you can see this red color in the top right corner, and that is the environment in the Arctic. That is the environment that is changing most rapidly on the planet Earth, where temperatures are increasing dramatically. And it's easy to feel a little bit frustrated and sad about this development, but we can also try to turn it around and try to see, well, what can we learn from these environments? Can we learn something about what is going on in these areas that are most affected? And can we take that knowledge to make predictions about what will occur in the global oceans in the future? But when it comes to picophytoplankton, the predictions are that in the future there will be more picophytoplankton than there are right now. So there's already a lot of picophytoplankton, but in future scenarios, with climate change, the total picophyton biomass is likely to increase further. And in this publication, that was published a few years ago, you can see that the areas where there's a prediction of the higher changes in the Picophytoplankton biomass is in the Arctic. So this brought me to participation in an expedition called the Synoptic Arctic Survey, which took place in 2021, where there was an opportunity to, in a very holistic way, study the Arctic and what the ongoing transformations would have, how that would be affecting the future oceans. So this was really an extremely large effort with 10 different vessels involved from different countries. And the focus on biology was very, very high. This is not typically the case for Arctic research. Much of Arctic research has focused on the dynamics, on meteorological or physical characteristics, but the biology in the Arctic is severely understudied. So the Central Arctic Ocean is an area that is covered in ice both winter and summer, and it's an area about 7 times the size of Sweden. So this is really a large area. But the really rapid temperature changes that are occurring in the Arctic is causing a change in the environment. So the permanent ice cover is changing to a seasonal ice cover. And the stratification and the nutrient availability is changing for phytoplankton communities. And this is then affecting the biological production and the distribution and composition of species. So when I went to the Arctic, I had 2 hypotheses about Picophytoplankton. So my first hypothesis was that the phytoplankton community in the Central Arctic Ocean was dominated by Picophytoplankton. And the second hypothesis that I had was that the cyanobacterium Synechococcus would become more abundant and increase its distribution into the Arctic Ocean. So with increased temperature, Synechococcus could also come into the Arctic. And what we found in our data was that there was very low chlorophyll in the Central Arctic Ocean. So the chlorophyll content was consistently below 1 microgram per liter. And this is quite different compared to what we see here in the Baltic Sea, where we typically see between 1 and 5 micrograms per liter. So it's a very low productive environment. And we measured chlorophyll A in 2 different size fractions, and we did find that Picophytoplankton had the largest contribution. So this is just an average of all of the data points that we had. It's consisting of more than 80 different data points. And the Picophytoplankton fraction contributed to 75 to 80% of the total phytoplankton biomass in the Central Arctic Ocean. We found chlorophyll max between 20 and 40 meters. And here on the bottom is just showing you the percentage of the total chlorophyll in the small size fraction. So you can see that this red color, which is more than 80%, it's dominating most of the surface waters. And it was the picoeukaryote Micromonas that was most common, and it was present between 2,000 and 20,000 cells per milliliter. And this is a picture of Micromonas. So this is the dominating picoeukaryote in the Arctic. I would also argue that it's the dominating phytoplankton in the Arctic, in the central Arctic Ocean. So this was an example then of a changing Arctic ecosystem where we found very low rates of primary production, and there was a higher proportion of picoeukaryotes. And with the changing Arctic Ocean, this would mean new habitats and also the possibility of novel species reaching the Arctic. So my second hypothesis was about Synechococcus in the future central Arctic Ocean. And there was a study from 2016 where they documented Synechococcus in a transect north of Svalbard. So I thought that, well, if I go 6 years later, maybe Synechococcus has really expanded into the Arctic. But from our results, we didn't find any detection of Synechococcus in the central Arctic Ocean. So we did a transit sampling from Helsingborg up to north of Svalbard, and we found that north of 76 degrees north, Synechococcus were completely absent. And this was both in molecular data set and flow cytometry. So we found no evidence of increasing Synechococcus in the Arctic yet. But if we think about the future, what could happen? If there is a shift towards more Picophytoplankton and this box of Picophyton biomass becomes larger, then the transfer into the trophic food chain can become less efficient. And what is typically thought about this picofraction is that it's not very rich in nutrients. It doesn't contain many essential fatty acids that is really needed for sustaining healthy ecosystems. So a shift towards smaller phytoplankton could mean more energy losses and less nutritious food for higher trophic levels. And this could then have very large consequences for the marine ecosystems. So I'm going to end this presentation with this sunset in the Arctic, which was the first sunset that we saw in more than 30 days. But we also have this very beautiful kind of chubby and fat polar bear that came to visit us. And I think this is also giving us hope about the future and how we as scientists can learn about our planet Earth and learn how to take care of it and also spread that knowledge among us. And I hope that that is something that you can take with you as you go out in the world and continue to study marine microbial ecology. And I also want to thank all the fundings and all the fantastic people that have contributed to this research. And thank you again for the invitation.